now, and it gives me great pleasure to welcome Jennifer Powers, who is Professor in the Department of Ecology, Evolution and Behavior, and Plant Biology at the University of Minnesota. And she received her PhD in biology from Duke University and then completed her postdoctoral studies at the State University of New York, Stony Brook, and the University of Minnesota. And she, as well as all her academic research, which you'll hear about now, she's also editor of the journal Biotropica. So thank you very much, Jennifer, for agreeing to talk to us, and over to you.
Thank you so much, Edvinder, for that introduction. And I have to preface this by saying I sort of have two half computers instead of one whole computer. If I drop out, I will immediately come back. I really apologize. And I want to thank Edvinder and all of you for your attendance. And I'm really excited to speak to you guys about the Bosque Seco for the next hour or so. I could, if you know me, you know that I could speak about the dry forest for days on end, but I assume Edvinder's just going to pull the plug after a couple days. But first, I just want to say, one of the other hats I wear is that I have the great pleasure of being the editor-in-chief of Biotropica, the journal of the ATBC. And so I, as Emilio Bruna, who was the editor-in-chief before me, always said, send us your best stuff. I hope to see all of you in person, and I hope to see all of your manuscripts. Okay, so we're going to spend the next hour or so talking about the seasonally dry tropical forest. And I should have added, just add water question mark, not a parentheses. So we'll see. I think that most of us, and certainly most people who aren't biologists, have some idea of what a tropical rainforest looks like. And indeed, I got my start working in tropical rainforests in Costa Rica and other places. But it is surprising for some people, even some people who are ecologists, to learn that not all tropical forests are rainforests. These are just examples of the diversity of forests that we call seasonally dry tropical forests, including these in Berenti, Madagascar. So I'd like to start by thinking about, what do we know about tropical dry forests? Well, we know that they have high biodiversity, and often the species that live in these forests are endemic. So they have high diversity and they have lots of endemism. We also know that they have high functional diversity. So if you look at this forest in Costa Rica, you can see evergreen species, deciduous tree species, and here brevideciduous tree species. We also know that these forests provide livelihoods for millions and millions of people. They provide lands for agriculture, opportunities for tourism, and provisioning of water resources and fuel wood. We also know that these are seriously threatened ecosystems. They're threatened by conversion to alternative land uses like agriculture. They're threatened by invasive species. They're threatened by drought, and they're threatened by fire. So now I want to turn our attention to what we don't know about seasonally dry tropical forests. This, of course, is a Whitaker climate diagram, and this is an attempt to divide the surface of the terrestrial biosphere up into different biomes based on climatic data. So these are probably pretty familiar to all of us, and I just pulled this one off the web. And if you go, I don't know if you can see my cursor. Yes, good. Okay, if you go along the horizontal axis, sort of leftward, you're decreasing in mean annual temperature, and up the vertical axis, rainfall increases. And what this shows is that we have a pretty good understanding of where we would expect to find tundra under very dry and very cold conditions. We have a good understanding of where we would expect to find tropical rainforest, where it is very warm and very wet. But when rainfall decreases, you can see here we have a much less precise understanding of what we would expect to find in this area where it is both warm but also dry. So this, in this figure, this figure lumps tropical dry forests or seasonally dry forests with savannas. Some of this, so to summarize, we don't even have a really good understanding of the climatic conditions under which we would expect to find dry forests. Some of this confusion, I think, was resolved in a paper by Kyle Dexter and his colleagues that came out in 2018. And what they did was to classify forests into different types: moist forests, semi-deciduous forests, dry forests, or savannas, and then actually just map out the climatic conditions under which dry forests occur. And you can see here that dry forests occur under conditions that are much drier than moist forests. So they have lower annual rainfall, and they also have a seasonality to that rainfall. And Kyle and his colleagues, they summarize with, I think, this really nice quote: the combination of seasonal drought stress and lack of fire leads to ecosystem function in dry forests that is markedly different from savannas or moist forests, which justifies their distinction as a unique biome. And this is a really great paper. I think it summarized a lot of important things, but I think it's also somewhat disheartening that, in 2018 is when this paper was published. And I would submit to you that you could not publish a paper in 2018 that questioned whether the boreal forest is a thing, right? We all agree the boreal forest is a thing. I think this just underscores how little we actually know about seasonally dry tropical forests, that, up until 2018, we're still having this debate. Is it a thing? Is it not a thing? So with that background, the goals of our lab are to understand the patterns and the mechanisms through which seasonally dry tropical forests respond to changes in land use and climate from microbial to biome-wide scales. And we do this through a number of approaches or conceptual approaches, through the approaches of community assembly, functional ecology, and ecosystems ecology. And we use a number of tools, tools such as data synthesis, large-scale experiments, long-term observations, and trait measurements. And so what I want to do today is just walk you through 4 topics where we've been working most recently, and really try to ask the question, what patterns do we see in the structure and function of tropical dry forests? And this is sort of a side note, but much of the research we do in my lab is in close collaboration with David Medvegi. He's an ecosystem modeler at the University of Notre Dame, and we work really closely with David so that all of the field data, my lab is really sort of the, we're the tree huggers, we're the hole diggers, we're on the ground, but we design our experiments and our data collection so that they're informative for simulation models. And collectively, our broad goals are to better represent seasonally dry tropical forest ecosystems in simulation models. So today what we're going to do is focus on some of our field data and take a closer look at the dry forest biome. And so I've divided this into 4 topics that I want to cover. The first is talking about a plant family that is abundant in the seasonally dry tropical forest of the Americas, the Fabaceae or the legumes. And then I'll talk about dry forest and how dry forest species respond to acute drought. And this will be focusing on our work in Costa Rica. The next thing I'll do is try to expand this understanding of hydraulic traits to a number of different seasonally dry forests. And I'll conclude by talking about soil biogeochemistry. And if there's time at the very end, what I'll do is tell you guys a bedtime story. I know many of you are in the UK and it's getting on, so I'll tell you guys a bedtime story if there's time. So this is a visual overview of the 4 different parts of the talk. We'll start with legumes, we'll move to drought, then we'll talk about tree hydraulics across the dry tropics in the Americas, and finish up with soil biogeochemistry. So legumes. Legumes, or plants from the family Fabaceae or Leguminosae, are interesting because they're abundant in seasonally dry tropical forests in the Americas. These are data from our long-term studies in Costa Rica, where about 18% of the stems that we measure in our forest plots across successional sites, across edaphic conditions, are members of the Fabaceae. From a biogeochemical standpoint, of course, Fabaceae is an interesting family because many of the 19,000 plus species can fix atmospheric nitrogen through symbiosis with rhizobia in nodules in their roots. So one of the questions that we thought about was, we think that legumes are important in seasonally dry forests, but we really lacked a comprehensive vision of the relative abundance of Fabaceae across environmental gradients in the Americas. And so this was a project led by a postdoc in my lab, Maga Hay. And what we did was to collate data from chronosequences. So each one of these green dots represents a chronosequence. It's a series of plots that vary in forest age. So these are all secondary forests recovering from agriculture or grazing. And these chronosequences are arrayed across a precipitation gradient. And our goals here were to identify the patterns of legume abundance across successional gradients and then across rainfall gradients. And we did this work in collaboration with the Second Four network. So what we have plotted here is forest age and the absolute basal area of legumes. Each one of these dots is a plot. Each one of these lines refers to a chronosequence. And we split the data, we split the chronosequences by climatic regime. We considered dry forest as forests that had lower than 1,500 millimeters of rainfall, and wet forest as forests that had above 1,500 millimeters of rainfall. And what you can see here, and the inset figure is the summary of these lines. So it's the mean values. You can see that on average, we find more legume basal area in forests that are older. So that tells us something, that succession's working. But we also see, if we look at the inset, that for any given forest age, say 50 years or 100 years, we find more basal area occupied by or accounted for by legumes than in the dry forest compared to the wet forests. This plot here takes the same data, but expresses the basal area of legumes relative to the total basal area in each forest plot. So it's a relative measure of how abundant legumes are in each one of these plots. And the inset here is highlighted in red. And so if we ask the question, where do we find legume trees across forests in the Americas? The answer is we find them predominantly in dry forests, and we find them particularly in early successional dry forests. And so this was a nice study in that it really gave us a comprehensive view, a comprehensive look at where we find legumes across the Americas, but it didn't tell us about the mechanisms that account for these patterns. We like to say that legumes, they're not just different, they're better. And there's many potential explanations that have been advanced to understand why legumes seem to be dominant or why they seem to be distinct compared to species of other families. One potential explanation, of course, is the observation that they have high foliar nitrogen. Many of them, of course, have the potential to fix nitrogen. This could give them the ability to have high rates of photosynthesis. Another mechanisms that we put forward in our synthesis paper was the idea that many of these species have bipinnate leaves, and they have very, very small leaflets. This might give them an advantage in dry or arid habitats. Other people have collated data and suggested that legumes have high water use efficiency, and these mechanisms aren't mutually exclusive. But what I want to do now is talk briefly about some studies we've been doing to suggest yet another potential explanation, which is that legumes might have a different regeneration niche in the dry forests, at least. So we work in Costa Rica mostly. We work in the Pacific Northwest part of Costa Rica in the province of Guanacaste. A lot of the forests are successional there, and a lot of them have lots of legumes. So we did a really simple study, and the goal of this study was to compare the germination dynamics of species of legumes compared to non-legumes. So we used 9 species of legumes, and we compared them to 23 species of non-legumes from diverse families. So here we've plotted date, and cumulative % germination is plotted on the vertical axis. We used different treatments that we hoped would help to facilitate germination. So all seeds were subjected to either one of 2 treatments. Submersion in boiling water, which we hoped would break the seed coat, or just a soak in water overnight prior to planting them out in these flats. So the different treatments here, the legumes are in orange, and the treatment with the seed coat break is in the light orange. The legumes that receive just the water soak is in the dark orange. The non-legumes are in the green colors, with the seeds that receive the seed coat break in green, light green, and the seeds that were just soaked in water in dark green. And the important points to take away from this graph is that across all these species, the legumes germinated quickly and synchronously. Within the first 10 days of the experiment, legume germination rates were 50% of the totals, compared to the non-legume taxa. So to reiterate, for these dry forest legumes that we looked at, they seem to have high germination rates and synchronous fast germination rates. And we've done other studies to compare the early growth of legume seedlings compared to non-legume seedlings. This was a shade house study in Costa Rica, where we compared the early growth and dynamics of 8 species of legumes and compared that to 14 species of non-legumes. And we did this under different light conditions and different soil conditions. And what we found here, so the different soil and light conditions are symbolized in the by different symbols, and the different the legumes and the non-legumes are indicated by the orange color for legumes and the green color for non-legumes. And what we've done here is just plot one of the metrics that we measured, height growth. So this is tracking height over time, and what you can see is that legumes, they germinate really quickly, but they grow really quickly too. So they grew quickly initially, and they maintained that height growth advantage weeks or months into the experiment. So let's try to put this in an ecological context. These are monthly rainfall data for where we work, Santa Rosa, Costa Rica. And what you can see is that rainfall is very, very pulsed seasonally. There's a very wet season and a very dry season. We think that this strategy of germinating quickly once the rains start and growing really, really fast may be a strategy that is well suited to this environment where rainfall is so seasonally pulsed. So germinate really quickly, grow like hell for 6 months. That seems to be a good strategy in this environment in Costa Rica. So the take-home messages from this part of the talk are that legume trees are more abundant. If we look across this, the Americas, legume trees are more abundant in early successional dry forest. And there are many potential mechanisms or hypotheses that might explain this pattern. But one of these mechanisms or hypotheses is that dry forest legume species, at least in Costa Rica, they seem to germinate quickly, they germinate synchronously, and then they grow very, very fast. Okay, so that was legumes. Now I want to switch gears a little bit. I'm still talking about Costa Rica. And think about these forests, these seasonally dry tropical forests in northwestern Costa Rica and their vulnerability to drought. Okay. So this, before the pandemic, I would say this was the question that kept me up at night. Now there's so many things to keep me up at night, like you guys. But this question is, will seasonally dry tropical forests be sensitive or resistant to future changes in rainfall regime? And before we started this work, I'd heard it argued both ways. I've heard people say, hey, dry forests are going to be extremely sensitive to changes in rainfall regime because these are tree species that are at their hydrologic limits. They're limited by water, and they're going to be very vulnerable. I've heard other people, different people say, hey, relax, seasonally dry tropical forest species are adapted to seasonal drought. These are species that are going to be extremely resistant to any changes in rainfall regime. And when we started this work, I was really agnostic. I just thought, hey, I just want to know which is it. Are they sensitive or are they resistant? And so what I want to do now is share some tree mortality observations that we have, which shed light on this question. So the questions we wanted to ask in this project were, is drought linked to mortality? Which species are vulnerable? Do traits help explain this vulnerability to drought? And then can we generalize to other forests? And this is a little bit of a spoiler alert here. These trees in the background are trees that have died because of drought. So we work in Santa Rosa, Costa Rica, in the Área de Conservación Guanacaste. Again, these are the long-term rainfall records from Guanacaste. You can see a number of things. One is that there's a lot of inter-annual variability in rainfall. Some years they get over 3 meters of rainfall. Some years rainfall is considerably less. But 2015 was the lowest rainfall year on record. You may recall that was the extreme El Niño event. And it resulted in the lowest rainfall record, the lowest rainfall year on record for Guanacaste. We wanted to know, is this drought linked to tree performance or tree mortality? Unlucky for us, we've been measuring demography in 18 permanent plots since about 2009. And so those years are indicated by the green dots here. And again, 2015, this extreme drought is indicated by the orange dot. So our first question was, is mortality somehow linked to low rainfall? So we have these 0.1 hectare forest inventory plots. We have 18 of them. We love them all. And we've been measuring recruitment, growth, and mortality of trees over 10 centimeters, again, since 2008, really, is when we started. Here we've plotted the average mortality rate for plots in the yellow bars, and this rainfall in Santa Rosa in blue. And what you can see from this plot is that, on average in these plots, I should say, are arrayed along successional and edaphic gradients. On average in these plots, about 1 to 3% of the trees die each year. By contrast, during this extreme drought in 2015, the mortality rates increased. They doubled to tripled. And mortality remained high in 2016, even though rainfall levels returned to more sort of normal rainfall. So I would argue here that this provides strong evidence that rainfall is linked to tree mortality, or drought is somehow linked to tree mortality, at least in this forest in Costa Rica. We had data. Our next question was, how does this mortality distributed among species? Are certain species more vulnerable or resistant to this extreme drought? And so we had data for about 53 species. For each of these species that are arrayed here along the horizontal axis, we had between 26 and over 200 individual trees per species to estimate mortality rate during the drought. So this is drought mortality rate plotted on the vertical axis. The average plot level mortality rate is in the dots, and the dashes here refer to the 2015 plot level mortality rate. And what you can see here from this figure is that, it's very clear some species are very vulnerable to drought. They lost over 35% of the individuals of a given species during this drought. And some species are very, very resistant to drought. They lost no individuals during this drought. And so our next question was, given that there's such a huge range of vulnerability to drought, what traits or properties of species might account for this huge variability that we see in response to drought? So the first thing we did was to use our large databases of what we call the soft traits, right? Wood density, specific leaf area, and we tried to correlate that species level mortality during the drought to those species level traits. And I'm not gonna bore you with these results because that really didn't tell us much. And so we needed to turn our attention to what people call hard traits, because they're much, much harder to measure. So we looked at these hydraulic traits, which we thought would be perhaps better indicators or correlates of species vulnerability to drought, because they're much more directly, these traits are much more directly linked to performance during drought. And so the first trait we collected data for was turgor loss point. This is the leaf water potential at wilting, or when stomata close. And we complemented these measurements with measures of the vulnerability to embolism. We used a method developed by Tim Brodrup here called the optical method of measuring P50. Essentially what you do is take a leaf and put it on a flatbed scanner. And as the leaf dries out, you make periodic measurements of leaf water potential at a different part of the stem. And you essentially just photograph embolisms as they are occurring until the leaf has completely dried out. And from these measurements, you can calculate the leaf water potential at 50% embolism. So this figure summarizes a lot of what we found in this study. This is a matrix of pairwise scatter plots with Spearman rank correlation coefficients between mortality rate during the drought, which is what I've plotted here in the top bar, versus all of the different traits that we measured. So the hydraulic traits, P50 and turgor loss point are here. And then we calculated the hydraulic safety margin. So this is turgor loss point minus P50. And we'll talk more about that. The soft traits of wood density and specific leaf area are plotted here: foliar nitrogen, foliar phosphorus, maximum height. And then we had a number of traits that describe the distribution of these different species along gradients. So we mapped out the distribution of species along a rainfall gradient, along a seasonality gradient, along gradients of dry season length or succession, and also looked for correlations among those variables. But I'm not going to talk about those because I think you can see pretty clearly from this that the main variables that we found that were correlated with the variability among species in mortality rate during this extreme drought were the hydraulic variables. Okay, so here what we've done is plot the mortality rate during the drought. Each one of these dots is a species, and the dot is proportional to the number of individuals we had to estimate that mortality rate. And we've plotted this against the variable that was best correlated with mortality during the drought, hydraulic safety margin. So hydraulic safety margin is plotted here, and we quantified that as turgor loss point minus P50. And so species, what this tells us is that species that had a very wide hydraulic safety margin suffered little to no mortality during a drought in comparison to the species that had a very narrow hydraulic safety margin. So what this figure does is break down that idea of hydraulic safety margin a little bit. This is taken from a paper Brendan Choate and colleagues wrote, and it traces two different curves essentially that you can make. The curve here in blue traces the water loss from stomata and cuticles, basically a measure of stomatal conductance as a function of increasing drought stress. So on this, on the horizontal axis, drought stress is increasing from left to right. And when you're measuring trigger loss point, essentially what you're doing is measuring this loss in conductance as a function of the leaf drying out. By contrast, when you're measuring P50, you're essentially tracing this red line. As a leaf is drying out, it is suffering embolism. So these are blockages, or these are air bubbles essentially that form in the vascular system, and they impair water movement. So in this red line here, it's tracing as drought stress increases, it's tracing the loss of xylem hydraulic conductance as a function of drought stress. So we can imagine for a given species that as for the species here, as drought stress is increasing, stomata are shutting down. At the same time, embolism is occurring. This species is shutting down conductance far in advance of when it might suffer loss of function or loss of hydraulic conductance. So we would say that this species would have a wide hydraulic safety margin. And by contrast, we can imagine another species indicated here by these dashed lines. So if we measure this, if we make the same measurements on this species, this is a species that we would say is more maybe acquisitive. It only shuts down its stomata after it has begun suffering loss of hydraulic conductivity. This species has a very narrow hydraulic safety margin. And when something like an extreme drought comes along, this is a species that's going to die. So the study in Costa Rica was novel in that it was one of the first studies where we had good data on forest demography, so we could calculate mortality rates. We had trees that experienced a large range of mortality under a naturally occurring drought, and we could link the vulnerability to that drought to traits like these hydraulic traits. So it was kind of, I was pretty sad when the drought happened, but I think we learned a lot from it. But this was just one way of looking at drought, and it's a way that relies on observational data. We've also taken more experimental ways to look at this question of how dry forest species and drought might interact. And we've done this by installing an experiment we call DROUGHT-X. What we've done is install these large-scale through-fall exclusion plots or exposures in plots in Costa Rica. So this experiment, we're using mixed species tree plantations that have 6 focal species. All the tree species are the same age. We know exactly how old they are because we know when they were planted. And the tree species we're focusing on represent different plant functional types in models. By working in plantations, we have easy access, and we've been able to cross our 8 through-fall exclusion or exposures with a fertilization treatment, so we can understand how drought and nutrients interact. So Herman Vargas is the grad student in charge of this, and he's working on our first 4 years of data that hopefully we'll have very soon. Okay, in summary, what I've just shown you is 2 studies in Costa Rica for our study site in Costa Rica. And what we see in Costa Rica is that these hydraulic traits predict or are correlated with the vulnerability of these different species to an extreme drought. You know, I feel like we learned a lot in Costa Rica, but that still leaves the question, how do these, you know, how can we generalize these results? Can we use these results to understand drought anywhere else? Can we extrapolate from our one study site in Costa Rica to other study sites across, or to other forests across the Americas? So this was the big question that I want to focus on next for the rest of the talk. This is the question: can we generalize to other dry forests? So to answer this question, we've been working with a series of collaborators in different dry forest sites. And the bigger goals of our project are to develop rich understanding of the biology of ecophysiology and biogeochemistry in these different dry forests that will help inform simulation models. This is a simulation model that colleagues Shangtao Zhu and David Medvigi and our group published in 2016, where we use plant hydraulic traits to define plant functional types for tropical dry forests, again, based on hydraulic traits. So a lot of the goals of the next part of the talk are really to try to extend this work from Costa Rica to other seasonally dry tropical forests. So this is the third part of the talk: DRYFQUR and hydraulic traits. So what we did to try to ask the question, to what extent can we generalize what we're learning in Costa Rica to other forests, was to develop a network with collaborators, which we're calling DRYFQUR. Why? Because we have 4 sites in Caxiaquihuic, Mexico, Guanica, Puerto Rico. Puerto Rico fell. Jabiro in the Interandean Valley of Colombia, and Magdalena Valley, and again our site in Costa Rica. And you may be thinking, hey, is this like a riff on Yadvinder Mali's Rainfall? Yes, it totally is. It's a riff on Rainfall. We only have 4 sites, and they're dry forest sites, so we call ourselves the DRYFQUR network. So here's a great paper from yet another network called DRYFQUR. And what they did was to use species records for plots across the Americas, and use this to develop, basically to bin plots and sites into different floristic regions. And so they used this approach to identify, I think it was 12 different floristic regions from Mexico all the way down past to the farthest southern extent of the dry forest. In our sites, we're sampling 4 of these floristic regions that were identified by the Banda et al. Paper. And these are some of our collaborators on the project. And we have great teams that we're working with in each of these sites. And this is work that was led by one of my graduate students, Herman Vargas. And again, the question here was how variable are seasonally dry forests in terms of the hydraulic traits if we look across these different sites. And so Herman visited these 4 sites. In each one of the sites, we had 4 small plots, and he collected a huge number of traits for about 102 species. And again, we focused on the traits that were harder to measure, like turgor loss point, like P50, like hydraulic conductivity. Not because it was fun, but because we thought that these would be the traits that would most reflect the behavior of these forests under different drought scenarios. So the first thing we did in this project was to collate the climate data for the 4 different sites we were working in. And so this just shows Colombia in blue, Costa Rica in green, Mexico in red, Puerto Rico in yellow, and it's different ways of looking at or summarizing climatic regime. What we can see here is that there's a huge variation among these sites in annual rainfall, in the seasonality of that rainfall. Our site in Costa Rica has high rainfall, but it's extremely seasonal. The site in Puerto Rico has very low rainfall, very long dry season length. So immediately this tells us that there's just a huge range of climatic variability across these dry forests, in addition to the floristic diversity that Banda et al. Discovered. Forest structure also varies across our sites. So these are frequency distributions of trees in different diameter classes across the sites. You can see in particular, the site in Puerto Rico really stands out as having lots and lots of very small stems. And this is not a succession of effect. This is, these are some of the oldest forests that we have in our network. So this is one of the figures, or one of the ways to look at the data Herman has been collecting. And what he's done here is plot one of these measures of hydraulic, one of these hydraulic traits, turgor loss point, for the different species that he measured in the different sites. And turgor loss point is measured in terms of leaf water potential. And so very large negative numbers indicate species that have a high drought tolerance. High values or less negative numbers indicate species that are not drought tolerant. And what we can see here, so the species are all plotted in the bar graph, and then the colored bar indicates the mean across species. What we can see here is that there is a large variability or large diversity of hydraulic traits within each forest. But then if we look across the forests also do sort out in terms of their hydraulic traits. And in fact, the forest that we work in Costa Rica, which has high rainfall and high seasonality, has a very low community weighted mean value of hydraulic traits. So if we look at turgor loss point, if we look at hydraulic safety margin, the Costa Rican forest has tree species that on average have a much narrower hydraulic safety margin or a much less negative turgor loss point or P50. And these are the forests that we would predict would be most vulnerable to an acute drought like the 2015 El Nino, which was indeed what we found. So these other forests, especially this forest in Guanica, didn't suffer the mortality during that drought that we saw in Costa Rica. And we've been looking at these data in other ways. This is a plot, this is a principal components analysis, where each one of these dots refers to one of the tree species that Herman studied. And the species are color-coded by leaf habit. With the semi-deciduous species in orange, the deciduous species in gray, and the evergreen species in green. And the question here we were interested in was whether or not tree species that have different leaf habits have predictable trait syndromes. So is there a sort of suite of traits that characterizes a deciduous species or an evergreen species? And I think what you can tell from this big sort of smear of points is that we didn't find very strong evidence that leaf habit separates out trees into functionally distinct groups based on the traits that we measured. So to summarize this, what we find when we expand our sampling of hydraulic traits across other across other seasonally dry forests, we find that two things. One is that within each of these forests, there is a range of different strategies. And if we think about hydraulic traits, and of course, then that that implies that there's also a range of strategies if we look across these forests. But I think it's encouraging that the community weighted mean values of these traits really are sorting out along these climatic gradients. Okay, so for the very last part of the talk, I want to talk about biogeochemistry. And I think I could pretty much summarize our results with this figure here. So this is work that was led by Bonnie Waring. She recently moved to Imperial College London from the United States. So our loss is your gain there in England. And Bonnie led this project. And what we did was to use our network and collect soil samples to get I would say, unprecedented view of the biogeochemistry within these forests and then across these forests. And the main question we were interested in identifying was, what are the spatial and temporal scales of variation in soil biogeochemistry across these sites? So again, we have our 4 DRYFQUR sites from Costa Rica to Mexico to Puerto Rico to Colombia. And within each of our DRYFQUR sites, we have replicated plots, and we collected replicated soil cores to 10 centimeters. And we repeated this sampling in the wet season and then in the dry season. And then we sent all of these samples, were shipped to Minnesota, and we measured basically every single thing you can measure in a Ziploc bag full of soils. We measured soil moisture, soil texture, particle size distribution, elemental composition, organic carbon, nitrogen, microbial biomass, phosphorus fractionations, nitrogen mineralization, basically everything. We measured everything we could. It took a couple years. And our predictions were based on this really iconic, I don't know if that's the right word, this really foundational paper that came out in 1989. And what this paper did was to look at the seasonal patterns of microbial biomass and measures of soil function for some dry ecosystems in India. They looked at dry tropical forests and savanna. And this is sort of a cartoon of what they found. Here, this summarizes, or it's sort of an icon to summarize microbial biomass. In the wet seasons, they found low pools or standing pools or stocks of soil microbial biomass. In the dry season, they found larger pools of microbial biomass. By contrast, the activity of the soil microbial community, which they indexed through net nitrogen mineralization, was much higher in the wet season and much slower in the dry season. And so they concluded that in the dry season, soil microbial biomass is sort of in a quiescent state. It's really in some ways sort of storing nutrients that are quickly mineralized, mobilized, and then available for plants once the wet season kicks in. And so this was our hypothesis or a prediction of what we would find. So this is a figure here that shows our 4 different sites. The different colors refer to the different countries. The stippling refers to each site in the dry season, and the solid bars refer to the same site in the wet season. And this is just one of the variables we measured, soil moisture. And this was reassuring because what we found was that soil moisture is higher during the rainy season. So we haven't quite, done a press release yet, but this did give us confidence because this was what we expected. By contrast, this is our measure of soil microbial carbon. So this is biomass carbon through fumigation extraction. And again, what we expected to find in Costa Rica, that the biomass pool is lowest in the wet season and highest in the dry season. But we found completely opposite patterns for Colombia, for Mexico, and Puerto Rico. Each one of these sites seems to be doing its own thing. So this is a figure here that summarizes sort of the next thing we did. Bonnie did a variance partition analysis so we could understand the dominant spatial or temporal scales of variability. So in dark black is the site. These are site, dark black color refers to variation that explained by the different 4 sites. The darker gray refers to variability within the plot at a given site. And then the light gray refers to variability within plot. So this is soil core to soil core variability. These are parameters that we only measured for 1 season of sampling. And you can see that pH, sand, clay, concentrations, these are more physical descriptors of soil variability, and they show this strong site signal. So they varied, the variation, the scale of variation was among sites. Oopsie, I'm going the wrong way. By contrast, this here summarizes many of the variables that we measured at a seasonal time scale. So here in white is the amount of variability that was accounted for by seasonal sampling. And the light gray, which is at the top bar and everything, are variables for which the patterns of seasonal variability differed among the sites. And so again, what you can see here, soil moisture had this strong seasonal variability. That's exactly what we were hoping. But really surprisingly, to me at least, many of the other variables we measured, things like fungal biomass, microbial carbon, dissolved organic carbon, microbial nitrogen, dissolved organic nitrogen, showed very modest amount of variability from season to season among all of these 4 sites. The next important, or the next interesting thing we found was that many of our measures of microbial function, like extracellular enzyme activity, which we use as one of our main metrics of function, showed lots and lots of variability within the plot. So the dominant scale of variability for microbial biomass, phosphorus, and these extracellular enzymes was really at the soil core. You know, what this suggests is that, like, if I were to measure enzyme activity in my front yard and my backyard, I would find just as much variability than if I measured microbial biomass in my front yard and wherever you are in a Zoom room. We found that these the dominant scale of variability for these, essentially for these nutrient cycling processes is at very, very fine spatial scales within a plot. And this hinders our ability to generalize across plots and across sites. So in summary here, the Bond et al. Paper was really lovely in highlighting the great floristic diversity within and among seasonally dry tropical forests in the Americas. I think our results for the biogeochemistry study that we did in 4 of these sites really highlights this large biogeochemical diversity that we find within dry forests as well. And these have implications, these patterns have implications for, for conservation. The Bond et al. Paper suggests that you couldn't conserve all of the diversity if you just focused on conserving one of these areas. I think our results for biogeochemistry suggest that you really can't extrapolate the results of a soil biogeochemistry study from Guanica to the dry forest in the Magdalena Valley. And so what's a way forward? Well, I think, similar to what Yadvinder has and the Rainfall network has done, is really to collect data along climatic, edaphic, floristic, and disturbance gradients to understand these patterns and then nest within that an understanding of mechanisms that differentiate or unite sites. So again, the take-home message here is that we found high spatial soil variability in biogeochemistry and much more modest levels of temporal variability when you look across these broad gradients, or seasonal variability, I should say. Okay, so this is it. In conclusion, I want to say the most important thing you should take away from this talk, is that tropical dry forests are awesome. These are just amazing ecosystems, but they're vulnerable to anthropogenic pressures, including things like drought. I think that what we lack is a comprehensive vision of what unites them and what differentiates them. If we think across the Americas, from Mexico all the way down to Brazil. But I think deriving this comprehensive understanding is really important for forecasting the future of these forests in a rapidly changing world. So with that, I want to thank Daniel Perez Aviles. He's my lab manager in Costa Rica, keeps all the balls in the air down there. I want to thank my funding sources and all of the collaborators, the wonderful collaborators who've made this work possible. And last, let's see here. Do I have time for a very quick bedtime story?
Yeah, go for it.
Okay, thanks, Yadvinder. Okay, so I know I come off as like being a champion for the dry forest, and definitely I fell in love with the dry forest, but my first love, of course, was rainforest. So I'm just like a pro forest person. And this is my love letter to the tropical rainforest at La Selva, Costa Rica. The sun was setting in a rainforest in Costa Rica. Parrots were squawking as they flew home to sleep for the night. A young coati named Pisotito was not sleepy, as he explained to his mother. Coatis sleep at night in the trees. After his family was asleep, Pisotito climbed down the tree to explore the forest. He was surprised to meet an animal he had never seen before. Her name was Paquita, and she was a paca. Pacas are nocturnal. They sleep during the day and are awake at night. Paquita took Pisotito to meet her friends in the frog swamp. He silently watched the jaguar from behind the trees. Day was breaking in the rainforest. Some animals like the kinkajou were getting ready to sleep, and some like the tinamou and the anteater were just getting up. Pisotito brought Paquita home to meet his mother. She suggested that Pisotito introduce Paquita to some of his friends. Pisotito's friends were diurnal animals. They were awake during the day, like the turtle warming herself in the sun by the riverbank. When the sun was high in the sky, they met a troop of spider monkeys and an eyelash viper. As day was turning into night, they said goodbye. They both thought about how the forest is full of interesting animals all day and all night long. That's the bedtime story. And I think the point here is that, you know, I think what we need to do is to take our love and our passion for the ecosystems we study and communicate it broadly to as many people as we can. So I'll leave you with that, and I look forward to our discussion.
Thank you, Jennifer. Do you want to stop sharing your screen? We can.
Yes.
And
there we go.
The risky strategy of reading bedtime stories to your audience at the end of a seminar.
Tell them a bedtime story. I figure if they're already asleep, it's no loss.
Great, thank you. I encourage anybody who wants to switch on their camera now. It's always nice to have a visual audience for this Q&A session. Okay, great. Thanks very much. There's a lot of really fascinating insights there. So we can open the floor to questions. I think, David, you've got a question there. Do you want to ask?
Hi, Jennifer, and thanks for a very interesting talk. Really liked it.
Thank you.
I was just wondering regarding the first part of your talk about legumes, about the role of bacterial or fungal symbionts in general, maybe in the role of in an indirect role behind the higher growth rates in the Fabaceae seedlings that you. So I was wondering whether there had been some sort of sterilization experiments to test whether actually these symbionts are key for this successional higher.
Yeah, that's a really good question. We haven't done that sort of experiment in our system, and we try to encourage a diverse microbial community in these shade house experiments. Other studies have looked at this question about whether or not the microbial community is limiting legumes versus some external thing like phosphorus. My guess is that you know, we've done these sort of experiments now in so many different shade houses that we always find this behavior of really quick germination and then very fast growth initially. And it's not entirely related to seed size, but you know, seed size explains some of that, you know, but not all of that. And I think that at that stage, the microbial community might not be as important as it, you know, as it is, you know, even after 2 weeks. In all honesty, when we started doing these, experiments, you know, once those legume seeds germinate, they grow 10 centimeters so fast. If you come back 1 week later, you've missed all the action. And so my guess is that's just, you know, something they do. And yeah, so I do think though the symbiont community is really an interesting sort of next step. And there's been some people who've done work in Mexico really sort of tracing mycorrhizal connections. So the Fabaceae, like many tree species, they have mycorrhizal associations, and doing network analyses to show that the acacia or mimosa, I guess, in this site in Mexico, are connected to many other species through mycorrhizae. So I think symbionts definitely have a lot to do with it, but I haven't dug down deeper.
Okay, thank you.
Okay, thank you. I'll just pitch in a question. All of your studies focus there on the neotropics, and how generalizable are these insights into both in terms of community structure, in particular, in the forests in Africa or
yeah, great question. And not having data from those sites, I think, or studied that up, I would be very reluctant to make those generalizations. And certainly I think at sort of a broader scale, I think what's so interesting about Southeast Asian is the dipterocarps and their reliance on ectomycorrhizal fungi, which can essentially act as decomposers and mine nitrogen. So is there, at a very broad biogeographic scale, a trade-off, from the Americas and nitrogen fixation and Fabaceae versus, dipterocarps with their ectomycorrhizal associates? Yeah, so I guess that's a way of saying I really don't know. I would. My dream, of course, and I'm on sabbatical this year, and my plan was to visit many different sites. I have a Fulbright, which who knows if I'll ever get to leave my bedroom here during the pandemic, but I'm definitely have plans to visit Tanzania, and I have a student who works in Madagascar as well, who's here. So I would love to dig deeper, so to speak.
Great, thank you. Ron, you have a question?
Oh, the question about the synchrony or asynchrony. Yeah, it's a good question.
Yeah. Hi. It was also along the lines of, I might have missed this in your talk, but you mentioned that the system is nitrogen limited, and that's why you see
no.
to be.
Yeah, no, you're astute. I didn't mention whether it was nitrogen limited or phosphorus limited. Two things. One is that we set up, Bonnie Waring, set up a large-scale fertilization experiment in our study site in Costa Rica in 2015. And so we have plots that are like 25 by 25 meters, and we add nitrogen or phosphorus or both to those plots. And what we've seen in those plots is that the main sort of flux or ecosystem kind of process that responds to fertilization is fine root growth, which we find is higher in our phosphorus plus plots. If we look at stem growth, what we see is that the legume trees seem to be responding more to phosphorus, and non-legumes don't show that degree of limitation. And I have a student now who's been doing a large shade house experiment in Costa Rica where we grew legumes and non-legumes under different phosphorus conditions. And what we're finding is that the legumes respond to phosphorus and the non-legumes don't. So my guess is that the legumes are phosphorus limited in that system, and the non-legumes are probably nitrogen limited or something else. And you had a question about the synchrony or asynchrony of germination. Yeah, so this is one of those things that I noticed when I used to take out. I spent several years living and working in Guanacaste in Costa Rica, and you'd always have to take your trash out to like the big thing where like it would be burned, and we walking through these like near the Guanacaste trees and all these different legume trees, and they would just have carpets of seedlings, and other species wouldn't. and I noticed this always when I was taking out my trash, I think that this forest in Costa Rica has such a defined wet season and dry season. And it used to be before, the sort of global weirding, you'd say, oh gosh, it's May 15th, the rains will come. There's like a very strong 6-month dry season where it basically doesn't rain at all, and then a 6-month wet season. And the timing of that wet season used to be much more predictable than it is now. But when it wets up really quickly. And I think if you're a seed and as soon as the rains start, you germinate quickly and then you grow very fast. I think that gives you a leg up, over species. In our germination experiment, we were really surprised that 2 months in, some seeds of not species that weren't legumes were germinating. If you're a seed and you're looking at a 6-month window to grow, and you only germinate, 2 months into that 6-month window, you're really losing out, I think, to put it in gross anthropomorphic terms. So I think that, if you have a very, very strong wet season, a very, very defined wet season, you're a seed, you germinate really quickly and you grow really, really fast, you're taking advantage of 6 months of the rains. If you germinate 3 months into that wet season, you're really, that's a seed that's missing out. Does that make sense?
Yeah, thanks for that. Yeah.
Yeah. Let's see, Jesus.
Thank you. Hi, Jennifer.
Hey.
Pretty cool work. Hi. I have one question coming back to basically with what you started in the presentation about this question about which would suffer the most, right? Which kind of forests, the wetter ones or the drier ones? But then you focus mostly on the tropical dry forest, obviously, because of vegetation. So I was wondering, based on the knowledge that you have gathered with all these experiments, like in the greenhouses and in the field as well, like if you would take this knowledge and also the knowledge that you have about the tropical wet forest, and if this forest would experience the same kind of drought with extent, let's say, or pressure, what are your thoughts about which one would suffer the most?
Yeah, so really good question. And I don't want to get myself in hot water here, but I would say that these, these hydraulic traits relate to help you predict a tree response to a drought in a way that that other traits don't. And so I think that my hope would be that we could take what we learn about hydraulic traits and then how that affects, growth or demography at a species level and make predictions about how a wet forest that suffers a drought, how those species should respond. So I think I think what this understanding does is really set this up as a testable hypothesis, right? As a prediction that we could use these traits in forests that we don't know to predict which species would be vulnerable to different levels of drought, I guess. I would use them more not as sort of a hypothesis we could go test than in a way to say like, hey, this forest, this forest is more vulnerable than that. I would think we can use them as a tool, I guess, is what I'm saying. Does that make sense?
Just to follow up,
Totally. Thanks.
just to follow up on that, across your DRYFQUR network, you showed that there was this broad plot mean correlation and plot mean traits. But did you find within individuals in the plot the same sort of relationship you found in Costa Rica in terms of mortality being predicted by?
We don't have the demographic data really for those other sites that we have for our site in Costa Rica. So the sort of linking that trait variability to demographics, we only have just sort of an anecdotal way to do that. And we're still, it's taken us a long time to sort through these data, and we only really have like the Herman's paper right now is under review. So what I hope is that we can build on these trait databases, but then also publish them and have them out there so people with demographic data can use them. Like our collaborators in those plots don't have the yearly data that we have for our sites in Costa Rica.
And was there a taxonomic structure to your hydraulic safety margin? Are there particular clades that are particularly vulnerable?
We haven't, I think Herman's on this call, but we haven't, like to my knowledge, we didn't, we haven't really looked at it in that way. Yeah, it all good directions.
Okay, great.
Directions.
Okay, Bill, you have a question?
Yeah, so does this hydraulic safety margin correlate with deciduousness? Yeah, is that it, Bill?
No, Bill.
Oh, no, that's
Ron,
someone
sorry.
else's question, but it's a related question. I was muted. So I'm wondering what the, if you could comment on the sort of why the hydraulic safety margin of leaves is a good predictor, given that so many of your species are deciduous. And you might think conversely to that, where having leaves that cavitate easily and therefore offer this hydraulic segmentation that would protect the rest of the stem. So could you comment on that?
Yeah, I can. So yeah, I think the bigger hydraulic safety margin, or the hydraulic traits we're measuring, don't map that well onto leaf habit. So leaf habit didn't, so whether or not a tree is deciduous or evergreen didn't help us much. Explain the variability and mortality among the 53 species in Costa Rica. And part of that, I think, is that, well, two reasons, I guess, that you just sort of pointed out. One is that leaf habit doesn't, so again, yeah, these hydraulic traits don't map directly onto leaf habit. The other thing is that, mortality is I'm probably presenting mortality as much more of a closed, like the factors that kill trees is more of a closed story, when there's been debate in the literature. Is it is it carbon starvation? Is it hydraulic failure? I think that that these may not be mutually exclusive. The thing we notice with some of those very vulnerable species, or the species that suffer high mortality, which the ones that are deciduous, is that they drop all of their leaves repeatedly during a drought. So they drop a cohort of leaves, they reflush. They drop a cohort of leaves, they reflush. And it makes you feel sorry for them. But I think it also, like, you're like, don't do it. Like, I think that mechanism of leaf habit as sort of leaf habit in this context might explain adaptation to a seasonal drought, but not adaptation to an extreme drought. And if that makes any sense. And the other thing, so I'm kind of painting a broad picture and glossing over some of the details. Some of the details in that site in Costa Rica are that not only was that 2015 El Nino low rainfall year, what really happened was it was a very short rainy season. So those rains didn't come until they were really like 3 months late, right? So I think that, so why is a leaf measure better than a stem measure? Well, leaf measure is what we had. Yeah, if that helps, I think. I guess that's where I'll, yeah, that's where I'll stop.
Okay, great, sir. Thank you. Ron, did that capture your question as well? Or is there anything else you want to follow on? Okay, that's right. Okay, no more questions in the chat. And any other questions? Feel free to just unmute and fire away if you have a sudden question in your head. If not, okay, I'd like to thank Jennifer again for a really insightful view of that. And it's really exciting that you're finding hydraulic safety margin is having such a predictive power in the sites, and that looks like real progress and possibly.
I'm glad we found something.
Yeah, because mortality is just so hard to predict in so many of our studies.
Yeah.
If that is a trait that really does have predictive value, that'd be a really powerful advance.
Thanks. And I want to thank you all for your time. And of course, like if you're interested, if you have further questions, you should feel free to email me. I love talking about seasonally dry forests. So thank you all for your time, and I'm really looking forward to interacting with you guys.
I suggest you unmute and we give an audible round of applause to our speaker.
Thank you all for your time.
Great, thank you. And we'll put this online so if anybody wants to look at it later or share it. Okay.
Nice meeting you, Vanessa, and thank you all. Thanks, Yadvinder. Thank you all.
Thank you.
Pura vida.
Pura vida.
Pura
Pura
vida.
vida.